So our next learning unit is on the citric acid cycle. So a little bit of a shorter learning unit here as compared to other chapters. So there's only a couple things that we'll go a little bit deep on, but for the most part, a little bit less dense of a chapter. We do go deep on kind of the structures and so forth in the actual metabolic pathway. And so glycolysis and the citric acid cycle represent two pathways that we kind of take a deep dive there. But not too much in this chapter in terms of regulation, but it also will set the stage for a lot of important biochemistry that's going to happen in the mitochondria. So our first slide here just kind of reminded us of where we've been, sort of a bird's eye view. Most of the chapters and learning units that we'll have here actually start with kind of this 30,000 foot view, if you will, kind of remind us of where we've been, kind of remind us of some important kind of overarching themes. And so one of the themes that's going to be kind of important here is to talk about disease states. So again, our important fuel equation here, talking about the utilization of oxygen, the generation of carbon dioxide, and truly this is the first time in the first metabolic pathway we're going to see a significant production, the majority of the CO2 that's produced as a function of fuel metabolism. So even though we've talked about other places where we generate and liberate CO2, it's really as we kind of go through the citric acid cycle here, and then pyruvate dehydrogenase, which is really the main process that happens to generate acetyl-CoA from carbohydrates, we're going to see some CO2 generation. So just kind of highlighting, beginning over here, we've got kind of an overview of the entirety of the metabolic pathways we're going to be discussing in Biochem 2. So again, we've studied glycogen metabolism in our last learning unit. But glycogen metabolism to glucose and then glucose via glycolysis to pyruvate. This really happens in an anaerobic state. It's rapid, but very inefficient, and again occurs in the cytosol. So those are those big picture concepts that I hope are sticking in your brain. The reverse is gluconeogenesis, again still happening in the cytosol, but that's where we take pyruvate, often derived from non-carbohydrate precursors, use it to synthesize the glucose that's needed. Again, highlighting here, these processes that occur in the cytosol, that occur in the absence of oxygen, is an important aspect to thinking about some disease states, particularly cancer. So one of the things that certain cancers are known to have hallmarks of are the fact that they like to live in this sort of anaerobic, hypoxic type of a situation. Again, without a requirement of oxygen, that means they can sort of subsist without the need to have sufficient mitochondrial function, and also can sort of subsist maybe without good oxygen delivery. So when we talk about tumors sort of growing and having a good blood supply, maybe they can subsist in a little bit of a weaker blood supply because they don't need oxygen in a way that other more aerobic tissues do. So again, these are the pathways that subsist exclusively in the cytosol in that anaerobic sort of state. We've already started to talk about dipping into processes that occur in the mitochondria, and this chapter really takes its first deep dive as to what's happening there. And so the first chemistry that we kind of talk about is the pyruvate dehydrogenase enzyme that's going to convert pyruvate into acetyl-CoA, kind of the first liberation of CO2. So remember when we talked about sort of the four main steps that we have of fuel metabolism, particularly thinking about carbohydrate fuel metabolism, is we have glycolysis to generate pyruvate from glucose. We've got pyruvate dehydrogenase to generate acetyl-CoA and CO2 from pyruvate. The citric acid cycle, which is going to fully metabolize acetyl-CoA into carbon dioxide, and then the electron transport chain followed by oxidative phosphorylation to kind of handle all of that energy that's been accumulated up to this point. We'll get to that in just a second here. But highlighting all of these processes are mitochondrial. The oxidation of fatty acids happens in the mitochondria. The full kind of metabolism of many amino acids requires enzymes that live in the mitochondria. Acetyl-CoA being again an important branch point metabolite, handling acetyl-CoA is a mitochondrial event. So one of the things that we're going to spend some time studying in our next learning unit on electron transport and oxidative phosphorylation, which occurs in the mitochondria, is the fact that many diseases are diseases of mitochondria. So if we kind of think of the four main kind of culprits or things that are kind of chronic disease or things that are problematic today, we've got cardiovascular disease, we've got Alzheimer's, we've got diabetes, and we have cancer. I think I said all four of them. So we've got cardiovascular disease, Alzheimer's, diabetes, and cancer. So again, those we're finding a lot of malfunctioning in the mitochondria. So speaking just a little bit more now about acetyl-CoA. We've talked about this already, but just kind of highlighting, because acetyl-CoA is made from pyruvate dehydrogenase, we're going to learn about those details in this chapter, but it's also derived from some ketogenic amino acids and from the beta oxidation or the breakdown of fatty acids. What happens with acetyl-CoA in the mitochondria is basically dictated by whether we're in a fed state or a need state. So again, acetyl-CoA, if we're in a fed state, is actually going to be synthesized into fatty acids in the cytosol. As we'll learn in this chapter, it's from the conversion of acetyl-CoA into citrate and the export of citrate into the cytosol. That's an important signal for a fed state, and so then we're going to synthesize fatty acids in the cytosol. Need state, well, again, if we need energy, we can take acetyl-CoA, put it through the citric acid cycle, 8 steps. Again, we're going to learn about the details of those 8 steps, again, mitochondrial, but we're going to generate these reduced cofactors. So our B vitamins are going to be harvesting those electrons. We're going to be generating the CO2 product that we have as waste. But again, we're going to talk about in our next learning unit what we actually do with these reduced cofactors in putting them through the electron transport chain, generating a proton gradient, which is then used to synthesize ATP via oxidative phosphorylation. So this learning unit, even though we've kind of talked about a lot here, this learning unit is really about the biochemistry that happens with the citric acid cycle. So another busy slide here, but again, a nice summary slide for us to think about. I do want you to know the main equation that we have here for the citric acid cycle. So again, it doesn't start with pyruvate. We're going to have the citric acid cycle start with acetyl-CoA here. We have another sort of question in your fundamentals that deals with the net reaction for pyruvate dehydrogenase. But we put it together sort of in this order. This is still the same equation that we have right here, but I like it written in this order a little bit better because it helps sort of keep track of what's going on. So we've got 2 carbons in acetyl-CoA. The net is that they end up becoming 2 carbons as CO2. So acetyl-CoA becomes CO2, and we've got electrons that we've harvested from that fuel oxidation. So those electrons go to 3 NAD pluses to generate NADH, and FAD to become FADH2. GTP and PI in what equates to a substrate level phosphorylation generates GTP. And we actually see the utilization of 2 molecules of water, and we'll see where those occur to that are just used up in the process. So we don't see those reflecting anything on the product side. So an important equation there. And when we think about the citric acid cycle, I want you to think about it like a circular clock reaction that we can split in half. Again, 8 reactions that occur in the mitochondria, and by definition we call these aerobic because they occur in the mitochondrion and they won't be committed there unless we've got oxygen available. So again, imagining we're splitting this in half, we're going to have 4 reactions that occur in what we're going to refer to as stage 1, and then 4 reactions in stage 2. So stage 1, I want us to think about these sort of like we thought about chemistry that happens in enzyme active sites. Remember the 3 steps for chemistry in an enzyme active site? We've got chemistry on the substrate, we've got to use all our reagents, and then we have to return the enzyme to its proper active state. So we're going to have that same philosophy here to think about the citric acid cycle. So again, the first half is really responsible for doing chemistry on the substrate. This is where we're going to see the generation of both molecules of carbon dioxide. We're going to see some energy production here, and this carbon dioxide generation with energy, we're basically going to see as decarboxylation reactions that occur via oxidation. So it's oxidative decarboxylation. And that's where we're going to generate, again, some NADH. So we can see 2 NADHs that are generated here in stage 1. What's important here is to distinguish 2 different kinds of molecules that we decarboxylate here. Isocitrate is sort of a hidden or a disguised beta-keto acid, so that decarboxylates fairly readily, so we don't need to think about any additional chemistry. However, alpha-ketoglutarate is an alpha-keto acid, and alpha-keto acids need help with decarboxylation. We've seen this before when we talked about the decarboxylation of pyruvate, which is also an alpha-keto acid. We need to incorporate TPP, which is derived from vitamin B1. So TPP is always going to be required when we see decarboxylation of alpha-keto acids. Not kind of written inherently on this slide, but again, pyruvate dehydrogenase chemistry, which really does decarboxylation, it is the same kind of chemistry, and so we're going to see vitamin B1 necessary there as well. So again, first half of the cycle is responsible for generating CO2, so chemistry on the substrate, and we do generate some energy that we see there. Second half is really about returning our whole metabolic pathway to an active state. And this is really about regenerating oxaloacetate, which is an important biological catalyst here. And the fact that it's catalyst has to do with a few things. Remember, catalysts are things that are used in a reaction but then must be regenerated. So we need to regenerate oxaloacetate there. So that's kind of an important piece to think about. Catalysts are something that are, again, going to accelerate the rate of a reaction. So having this kind of as part of generated in our 8th step and used in our 1st step is an important kind of distinction for that specific metabolite. So stage 2 really is about returning our system to its active state. So we begin with succinate here. So again, we've got our 5th step really converts succinyl-CoA into succinate. So this thioester here really being kind of a high energy bond is why we can think of this step as occurring via substrate level phosphorylation to generate GTP, but then we end up with succinate here. As I kind of joked around with you guys, succinate sucks in terms of having functional groups because there's not really any functional groups in having an alkane here. So the next 3 steps are going to deal with oxidation, hydration, and then another oxidation step. So we've got oxidation of succinate as an alkane to fumarate as an alkene. Then we're going to have hydration of that alkene to generate malate as an alcohol. And then we are going to have oxidation of that alcohol functional group to a ketone to regenerate oxaloacetate. So some things that we'll highlight again once we go through the details of each of these metabolic steps is when we see oxidation of alkanes to alkenes, we tend to see FADH2 being helpful in that chemistry. And when we see oxidation of alcohols to ketones or the reverse of that, that tends to be the incorporation of that vitamin B3, that niacin as NADH. Okay, so that's a nice overview of not only the entirety of the metabolic pathways that we've studied and are going to study, but now of thinking about an overview of our 30,000-foot view of just the components of the citric acid cycle. We're going to spend some time first kind of dealing with pyruvate dehydrogenase, sort of as a step 0, if you will, of the citric acid cycle, and then we'll delve into the 8 reactions of the citric acid cycle. So the first thing, again, we're going to talk about is pyruvate dehydrogenase. So this is the first time we've actually seen an enzyme that's going to operate as a multi-enzyme complex. So kind of shown up here, nothing that you need to remember, but it's just interesting to think about. Each of these little spheres represents an individual sort of enzyme that is in this multi-enzyme complex. So we can see that there's going to be 3 different kinds of enzymes. We see 3 colors here. We're going to refer to these 3 enzymes as E1, E2, and E3. So nature loves symmetry. So what's really neat is if you remember some of your Gen Chem or maybe inorganic chemistry, we see some symmetry here in terms of if we imagine a unit cell here, we can see that some of these kind of occupy edges, some of them occupy faces, and so forth. So when we put this all together, there's some beautiful symmetry in this multi-enzyme complex that basically has 3 kinds of enzymes comprising different domains. So I want to kind of highlight first here the chemistry that happens in pyruvate dehydrogenase. So again, an equation that you need to know for your fundamentals. Pyruvate is ultimately oxidatively decarboxylated into CO2 and acetyl-CoA. So again, that's what happens with the 3 carbons here. 2 of them stay on as an acetyl group on acetyl-CoA, and then 1 is liberated as CO2. So that's again why we need CoA to generate acetyl-CoA. And because this is oxidative decarboxylation, we must have some reductive process. So NAD plus is converted into NADH. So again, we can see here this is not going to be happening unless we are in the mitochondrion, because we're going to be using up an additional equivalent of NAD plus to NADH. So it won't happen unless we are in an aerobic environment. So just to highlight, we've got these 3 different enzyme sites. And so we've got E1, which is the vitamin B1 containing enzyme active site. So this again is going to be where we're seeing decarboxylation of pyruvate. Again, pyruvate being an alpha keto acid. We're going to see more clearly on the next slide here, but what happens is we take and we decarboxylate pyruvate, then we transfer those 2 carbons in this hydroxyethyl group to E2. Again, 2 steps to that process. Second enzyme active site that we see here is called E3. That's going to be kind of this long arm that's going to reach over, and it's going to basically deliver that acetyl group to CoA. And then we're going to have E3, which is kind of a regeneration type of an enzyme active site, which helps kind of get our enzyme back to its active state. So that hopefully will make a little bit more sense as we look at this picture here. So this again was a homework problem here, and I'm going to flip to and look at my homework here as I kind of highlight with question 3 for you guys, talking about what happens in each individual step, and then again if there's any cofactors that are needed, and then thinking about whether or not once we've done some chemistry that site is either ready, inactive, or busy, and how we can think about that in terms of regenerating the enzyme active site. All right, so pyruvate dehydrogenase chemistry. So pyruvate's going to come here. It's going to come into the E1 enzyme subunit. Pyruvate, being an alpha keto acid, is not going to be easily decarboxylated without TPP. So TPP is part of the E1 subunit. And so the first thing that happens in step 1 here is we are going to have the loss of CO2. So we have a loss of CO2 here from pyruvate, and then the other 2 carbons are transferred to TPP as that hydroxyethyl group. So if we think about what that means here, it means that E1 is busy because TPP is kind of holding on to that C2 group, and E2 and E3 are ready. We really haven't seen them be used yet. So if you're following along with homework question 3, E1 is busy, E2 and E3 are ready. So if we look at what happens in step 2, so now we've got E1 sort of holding on to these 2 carbons. What's going to happen is the long arm of E2, this lipoamide arm, is going to come over and it's going to grab these 2 carbon units from E1, and it's going to generate this acetylated arm, if you will. So we've just transferred this acetyl group. So what happens in step 2 is we've got an acetyl transfer from E1 to E2 that regenerates TPP in its free form. So E1 is ready. Its TPP is free, ready to kind of help out with another round of chemistry. But we've got our E2 group now being busy. It's holding on to this acetyl group. Moving to step 3 now, this long lipoamide arm, which has the 2 carbon group that's attached to it via this thioester bond, is going to do a trans-thioesterification reaction. Essentially, it's going to be transferring this acetyl group to another sulfur atom. Remember, CoA is really CoASH. So it's going to be transferring this acetyl group to CoA to generate acetyl-CoA, and in doing so, it's left in this reduced or inactive state. So if we think about what happens on step 3, again, it's an acetyl group transfer from our lipoamide arm on E2 to CoA to generate acetyl-CoA. Now the problem is we do not have E2 in its ready state. We're going to say now that it's inactive. Okay, it's inactive because it's in its reduced form. So E1 is still ready, and for the rest of kind of this process, it's going to be in its ready state. And E3 is also ready. It's not been used yet. So steps 4 and 5 are really about regenerating our enzyme active state. So we're going to have now in step 4, E3. So what really happens is E3 is going to regenerate this lipoamide arm. Remember, this is kind of a long arm that's going to dip back and forth between different enzyme active sites. So E2 now has, if you want to think of it like a little mangled arm here, it's not in the right state. So it dips over into E3, And there's going to be a disulfide exchange reaction here. So in this disulfide exchange reaction, we have this group here, which is in its oxidative state, does a disulfide exchange reaction with lipoamide's arm, which is in its reduced state. This lipoamide arm becomes reoxidized into its active state, and this group now becomes reduced. So again, if we were to kind of take tally of what's going on, E1's been active since we got rid of that C2 group on it. Now E2, which was formerly inactive because it was reduced, is now in its active state. It's oxidized, it's ready to do chemistry. However, E3, in helping to fix that problem, now itself has become reduced and is now inactive. So step 4 is really the regeneration of E2 by E3, but that unfortunately means now that E3 is in an inactive or reduced state. So the 5th step really now incorporates all of the materials that we need to have. We're going to see NAD+ coming in and being used, itself being reduced so that we can reoxidize this group that we have here in the enzyme active site. So questions 4 and 5 on your homework actually still build off of this, so I'm going to just kind of highlight where they are. One of the questions asks about the different B vitamins that we need. There's actually 4 B vitamins that we see in this process. 2 of them, which again are familiar, are vitamin B2, as anything that is flavin or riboflavin related, and then vitamin B3 for NAD+. Vitamin B5 is important for that CoA group, and then vitamin B1 for our TPP sync. So the other thing that was part of these questions, question 5 kind of talked about drawing and thinking about the different thiol groups that we have in pyruvate dehydrogenase. So we've got the thiol groups here that are present in this lipoamide arm. And so these are 3 carbon atoms that are here. So a unique sort of 5-membered either disulfide or thiol containing ring that we have here, or a ring open form. So that's kind of a unique chemistry that can toggle back and forth between reduced states and oxidized states. And what we have over here in E3, it's not that it is a 4-membered ring. These sulfurs actually are part of 2 cysteines that are fairly close to one another in a 3-dimensional space, so that they can actually disulfide link to each other and perform this disulfide exchange reaction. So these are cysteine residues that we have here. So again, that's the chemistry that happens in pyruvate dehydrogenase. What's really neat here is if you actually look at what that enzyme active site looks like. Remember, we kind of highlighted that this was this multi-enzyme complex that had a lot of symmetry. So what they're showing here is just kind of a surface map of all the different areas and zones. And what's kind of represented here is this long lipoamide arm. And what they're really showing is it dipping back and forth between different enzyme active sites to kind of do its chemistry. So kind of a neat structure affords function picture there. All right, so that was pyruvate dehydrogenase after sort of an introduction and a 30,000-foot view to the citric acid cycle. So now we're going to move on to kind of very similar to what we did with glycolysis, really looking and taking a deep dive at the structures and the functional group changes that happen within the citric acid cycle. So 8 steps that we're going to have here. You have a homework question that kind of goes through, question 6 kind of goes through sort of a summary table like we had for glycolysis, just reminding us of the different substrates, enzymes, products, and the different chemistry that's happening. So we're going to highlight that as we go through 8 slides here. Again, always trying to orient ourselves to where we are in this process. So just like with glycolysis, I want you to know enzyme names, enzyme step numbers, products, reactants, coenzymes that are needed. The whole exam won't be like that, but you are going to have some questions that ask you some detailed things about, what step generates CO2, what steps generate NADH, and so forth. So I'm going to walk through each one of these steps, kind of highlighting the important things that happen here. So citrate synthase, first enzyme and first step in the citric acid cycle. So again, because this is a cycle, there's not really a start or an end per se to it, but we have some chemistry that happens here to kind of do carbon-carbon bond formation. So this is kind of a unique thing we have at the very end of our learning unit here. We'll talk about mechanism for this, but carbon-carbon bond forming chemistry. We take the 4-carbon oxaloacetate, we condense it with a 2-carbon acetyl-CoA group. And then again, this enzyme happens, or this chemistry happens with citrate synthase. This is a spot where we're going to need to see the incorporation of water. And we generate a C6 citrate, and then we liberate free CoA. So just to highlight, sometimes you'll see CoA just written as CoA. I like always writing it as CoA-SH because that reminds us that there's a free thiol group there that can do chemistry. So looking just a little bit into the enzyme active site here, again, we've got oxaloacetate and we've got acetyl-CoA. We've got some amino acid residues that kind of help facilitate chemistry. We'll see this mechanistically at the very end here, but we're going to use acetyl-CoA to generate an enolate. And then that enolate is going to nucleophilically attack the carbonyl carbon here of oxaloacetate, and we're going to generate that carbon bond, and we're going to generate Citriol-CoA, so citrate that's still attached to CoA via a thioester bond. And this is why we need water here. We're going to hydrolyze this thioester much like you'd hydrolyze an ester, and we're left with a carboxylate group, and then our free alcohol, which is in this case the thiol, as CoA-SH. So again, no real vitamin cofactors that are necessary. We need to utilize water here. Important chemistry that happens as we form a carbon-carbon bond. And the purpose of this is we've basically loaded that 2-carbon acetyl group onto oxaloacetate to be able to be oxidized. So one of the things that we'll highlight as we go through this process, these 2 carbon atoms that come in from acetyl-CoA at the beginning are not the 2 carbon atoms that come off in our decarboxylation steps here. So that's an important distinction to make. We'll kind of highlight as we get there to it. All right, so first step, citrate synthase. Second step is called Aconitase. Some of these enzymes have weird sort of historical names, don't necessarily describe the chemistry that's going on the way that citrate synthase does. So always just kind of a neat little historical thing. If you have something that has sort of a little bit of an odd name, it's probably because it was an enzyme that was discovered and named before we had sort of a formalism for naming enzymes. So aconitase is an enzyme that's important for doing isomerization chemistry. So step 2 in the citric acid cycle is isomerization. So we take citrate and we're essentially going to move where this OH group is. This OH group is kind of in the middle here, and then we're going to move it to be down here. And the reason that we're going to do that, if you look at this region that we've boxed sort of in gray here, we've got, here's this carboxylate group that is going to be the first CO2 that comes off. This one down here is going to be the second CO2 to go off. I kind of highlighted this just a minute ago, but since it's pointed out here on this slide, these are the 2 carbon atoms that come in from acetyl-CoA. So you can see that those are not carbon atoms that come off immediately as CO2. The first CO2 that comes off is this one right here, and then the second one that comes off is down here. So why do we need to do this chemistry? Why do we need to do this isomerization chemistry? So what is the purpose of it? Even though the chemistry we can label as isomerization, the purpose is that we create a hidden beta-keto acid. Hopefully everybody can see if this is our acid group, not the alpha carbon, but the beta carbon has a hydroxyl group. So what we have here in isocitrate is a beta hydroxy acid. Well, a beta hydroxy acid is just an oxidation step away from being a beta keto acid. We know that beta keto acids easily decarboxylate, so what we're really doing in this step is setting ourselves up for the next step, which is to decarboxylate that beta keto acid. So we saw chemistry like this when we studied glycolysis, when maybe we saw that we were generating a high energy species because we knew the next step was going to be substrate level phosphorylation. The same thing sort of happens here. So aconitase interconverts citrate with isocitrate. The chemistry is isomerization, and the purpose is to create a hidden beta keto acid. Third step is the first step that we actually see decarboxylation. And when we see these decarboxylation chemistries that happen in the citric acid cycle, we're going to see them as kind of oxidative decarboxylation. So Oxidation happens first. Remember, we said we made isocitrate because it was a hidden beta-keto acid. We need to oxidize this hydroxyl group to generate that carbonyl. And so here this is shown as kind of a transition state, not necessarily an isolatable intermediate. Here we have our beta-keto acid, which easily decarboxylates. So we're going to remove this red CO2 group here. And then once we protonate, we generate alpha-ketoglutarate. So again, alpha-ketoglutarate is going to be the product of isocitrate dehydrogenase. It's 5 carbons. So again, this purpose of oxidative decarboxylation is to remove a carbon atom. So you're going to see throughout kind of some of these steps here, we're going to show the net of what happens with this chemistry is taking acetyl-CoA, and here's the products that we make. So this is not the net reaction. We don't have the NAD plus and the FAD on the left side, but we're going to talk about the products that we make, and we're just going to kind of tally up when we've started to make these. So this is the first CO2 that's generated, the first of 3 NADHs. So again, oxidative decarboxylation happens in the isocitrate dehydrogenase reaction. Isocitrate is converted into alpha-ketoglutarate. Fourth step, and remember we split our citric acid cycle into 2 4-step stages. So this is really the last step of what we're referring to as stage 1. We've got alpha-ketoglutarate dehydrogenase. So alpha-ketoglutarate is an alpha-keto acid. So again, now this CO2 is going to be coming off, but because we have an alpha-keto acid, we need to see TPP involvement. We need to see vitamin B1. We're going to refer to this as a pyruvate dehydrogenase homolog because all of the chemistry that we saw happen with E1s, E2s, E3s, all of that, the same thing happens with alpha-ketoglutarate. Still is oxidative decarboxylation, just like we saw the production in pyruvate dehydrogenase chemistry of NADH, and then we've got CoA attached to our product. In this case, we're going to have Succinyl-CoA. So when we take a carbon off of alpha-ketoglutarate as CO2, the 4-carbon piece that's left is essentially succinate that's attached via a thioester linkage to make Succinyl-CoA. So again, last step of what we're referring to as stage 1 is alpha-ketoglutarate undergoing oxidative decarboxylation in a pyruvate dehydrogenase-like enzyme to generate Succinyl-CoA. Important cofactors that are necessary is we need CoA, we need NAD+, and we need TPP. So we generate the same kinds of products that we saw with pyruvate dehydrogenase. We generate NADH and we generate CO2. So again, taking up a tally here now, we've generated at the completion of stage 2 both of the CO2s that are necessary, 2 out of the 3 NADHs, and again, the rest of these are going to come in stage 2. So the 5th step really begins sort of our process of regenerating oxaloacetate. So don't worry about the steps here that we've kind of shown here with Succinyl-CoA synthetase, but what we're really going to do in this step is we're going to take that high energy thioester bond that we have here with Succinyl-CoA, and we're going to use it to synthesize a molecule of GTP. So we can sort of think about this as sort of a hot potato of phosphate. So we're going to start with inorganic phosphate and then our high energy Succinyl-CoA intermediate. And we're going to essentially be transferring this phosphate around to a bunch of different groups to ultimately allow it to be transferred to GDP to generate GTP. So we can sort of think about this like substrate level phosphorylation. We're using a high energy intermediate to couple together inorganic phosphate and a nucleotide diphosphate, in this case GDP, to generate GTP. So again, the products of this reaction are we liberate free CoA, we generate succinate, and then one of our high energy species here that's generated as a product in GTP. So as we'll see as we finish out stage 2 here, succinate really is a molecule that sucks in terms of having functional groups, right? It's got an alkane essentially in this internal portion as a functional group. So what we're going to see in steps 6, 7, and 8 are the consecutive oxidation, hydration, and oxidation of one of these carbon atoms to return succinate to oxaloacetate. So again, if we think about the first step here, succinate dehydrogenase, we're going to see this alkane group be converted into an alkene. Anytime we see this functional group interconversion, we're going to see the utilization of FAD as sort of our redox cofactor. So vitamin B2 is going to be necessary for that. So we generate FADH2. At this point, we've actually generated all of the high energy species or all of the chemistry, kind of the waste products from the citric acid cycle. One of the things that's important to highlight here is succinate dehydrogenase is actually complex 2 of the electron transport chain. So even though this chemistry is happening as part of the citric acid cycle, it's utilizing a complex, an enzyme that's embedded within the inner mitochondrial membrane to do that chemistry. All right, so step 6 with succinate dehydrogenase does oxidation chemistry of an alkane to an alkene. Necessary cofactors are FAD, that's actually an enzyme-bound FAD that's part of complex 2 in the electron transport chain. So even though we generate this FADH2, it's not a free FADH2. It's an FADH2 that's shuttled immediately to the electron transport chain. 7th step here is the hydration of an alkene to generate an alcohol. Now we're starting to introduce some functional groups that we can see being reconverted into oxaloacetate. So fumarase is really just the hydration of fumarate to generate malate. Okay, so we need water to do that hydration chemistry. Nothing really changes in terms of the chemistry or the products that we're making here, but again, this process is important because we're regenerating oxaloacetate. So the last step is malate dehydrogenase. So again, this is an enzyme we've spent some time studying in lab. So malate dehydrogenase does some redox chemistry that converts the alcohol functional group in malate to the ketone functional group in oxaloacetate, and we regenerate that important metabolic intermediate, that branch point metabolite. Anytime we see oxidation chemistry that involves oxygen-containing functional groups, we're going to see NAD plus and NADH that's involved with that. So again, malate right now is going to be converted into oxaloacetate. That's an oxidation, so a reduction is the generation of NADH. So again, one of the things to highlight with this chemistry is that the delta G is large and positive. Again, that means that we make very little of this product. So it's, again, considered to be a catalyst. An important highlight here is that the oxaloacetate that we utilize for gluconeogenesis is not made by this process in the citric acid cycle. Remember, we'll see this in a few slides here, but pyruvate carboxylase is what we utilize to generate that oxaloacetate. We learned that in our last learning unit, but just highlighting, oxaloacetate has different sort of sources for where it's made. So a tiny little bit of oxaloacetate squirts out of this process, but because it's coupled to citrate synthase, we can pull this reaction forward. We saw that same kind of reaction coupling with reactions 6 and 7 in glycolysis. Remember, reaction 6 had a positive delta G, so we could pull that reaction forward with the very exergonic reaction 7. So at this point, we've regenerated oxaloacetate. We've completed one round of the citric acid cycle. Just mechanistically to kind of look at what happens here with the chemistry with citrate synthase, this is really, again, a Claisen condensation reaction. So we're going to have an enolate that we react with a ketone, and that enolate is derived from an ester. So again, here we've got an ester. Imagine this being a thioester, for example, acetyl-CoA. So again, if this was acetyl-CoA, this would be a CH3 group here, but remember, the alpha carbon is slightly acidic because we can generate a resonance stabilized enolate. So we're seeing this resonance stabilization here. Ideally, the electrons will sit on that oxygen because it's more electronegative, but we want to have the nucleophile really being our carbon atom here. So we're going to reform our bond here, nucleophilic attack now on our ketone here, and if this ketone is part of oxaloacetate, that's how we're going to be forming our bond between that C2 group from acetyl-CoA, and then we're going to have here our carbonyl group in oxaloacetate. Nucleophilic attack generates a tetrahedral intermediate here. So here's our tetrahedral intermediate. When you do chemistry on a carbonyl that's either an aldehyde or a ketone, the only option here is to protonate that tetrahedral intermediate because you don't have any good leaving groups. And so protonation of this is going to generate our beta-hydroxy acid, or beta-hydroxy carbonyl containing compound here. In this case, it's going to be a beta-hydroxy ester. Again, don't want an ester, you can hydrolyze it away. That's why we see water being required at citrate synthase, because we're going to hydrolyze off that CoA, that thioester. It's going to be hydrolyzed there, and we generate our free carboxylate group. And in this case, our beta-hydroxy acid is going to be citrate. So the last little piece to talk about here is regulation. And so we are not going to take as much of a deep dive here with regulation with regards to enzymes in this metabolic pathway like we have done for others. But I'm just going to kind of highlight a couple of things here. The first is thinking about how we sort of regulate pyruvate dehydrogenase. And this is really important because pyruvate dehydrogenase is really sort of seen as the gatekeeper for aerobic metabolism of carbohydrates. Because remember, pyruvate dehydrogenase chemistry is not reversible. So if we have committed something via pyruvate dehydrogenase to acetyl-CoA, we're sort of committing it to chemistry happening in the mitochondrion. So I'm not going to say too much about regulation here, other than highlighting that it is something that is regulated by phosphorylation, so covalent modification via a residue on E1. So again, we've got an E1 here that could either be cycled between a phosphorylated or a dephosphorylated state. So again, if we want to think about pyruvate dehydrogenase being active, we talked about being active in a dephospho or fed state. So this is sort of consistent with our logic check here, that we want pyruvate dehydrogenase to be active during a fed state so that we can kind of handle the accumulation of pyruvate. So we're going to have a phosphatase actually being responsible for removing that phosphate, and then a kinase again for adding that phosphate. So I'm not going to talk too much right now about pyruvate dehydrogenase regulation. One of the things that is sort of interesting that we'll see pop up is it's not uncommon to see a phosphatase that is going to be doing some chemistry being PP1, and then a kinase that's doing some chemistry being pKa. So not the case here, but again, cascades can lead us to have pKa being involved in this process or PP1 being involved in this process. So again, not going to say too much about that here, but I do want you to understand that pyruvate dehydrogenase is subject to covalent modification, and that covalent modification is on E1. We also kind of took a little bit of a look here, and there was a homework question sort of relating to how we think about regulating the citric acid cycle enzymes themselves, so those 8 enzymes. And so we just kind of went through and used the same philosophy that we've talked about before, with key regulation points are going to be reactions that are far equilibrium. So we can see 3 that we have here, reactions 1, 3, and 4, so citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. So those again are going to be flux control points. When we applied our logic here of either eliminating or adding or including an earlier or a later step, because this is a cyclic process, that logic doesn't necessarily apply as much because it's a circle, so there really is no start and no end. So these are 3 points that are subject to regulation. We're not going to go into the details for how they're regulated, but I will comment that one of the important metabolites that's important for regulating is NADH. You can see that NADH feeds back and inhibits many of these processes in which NADH is generated. So one of the things that's going to be important to think about is the ratios that we have between some of these redox factors in their oxidized or their reduced forms. And so again, if we think about having a high sort of concentration of NADH, that's going to mean that we've got a lot of sort of reduced cofactor, probably more of a fed state system. So we kind of need to be upregulating processes that are going to handle that. And we want to slow down processes that are going to be generating it. So again, we're going to slow down processes that generate NADH, like the citric acid cycle, if we have a high concentration of NADH. So if we look at this NAD plus to NADH ratio, it can really be telling us whether we're more in a catabolic or an anabolic state. So one other thing to just kind of highlight is reminding us again about this large positive delta G that we see from malate dehydrogenase. So again, that means we're going to favor our reactants, not our products. We're going to have a very low concentration of oxaloacetate. And again, satisfying the definitions for a catalyst, oxaloacetate is going to help make this reaction faster. It's used up and then regenerated, so we can see used up in the citrate synthase reaction, regenerated in the malate dehydrogenase reaction. And again, present in very small amounts, which by definition is what we see for a catalyst. So not a ton in this chapter talking about regulation. So the last kind of lecture that we had here on the citric acid cycle was kind of thinking beyond the cycle and thinking about kind of siphoning off or siphoning in, or variants of kind of chemistries that happen here. So we spent a little bit of time talking about both ana or cataplerotic reactions. So anaplerotic reactions are going to be things that basically add to the pathway. And so these are going to be things that kind of siphon into this pathway. So the green that we have here. Cataplerotic reactions, you can think of that being like cannibalizing the pathway. So we're going to be taking things off. So two that kind of dealt with this, and this was homework question 12 that we had here. We're kind of dealing with three pathways, both kind of starting with pyruvate, kind of dealing with both cataplerotic and anaplerotic reactions here. So with this green pathway here, we can kind of see, and this is question B, 12B on your homework, we can see in this green pathway, we have pyruvate being made into pyruvate carboxylase. Remember we talked about we needed to have a different way to make oxaloacetate. Malate dehydrogenase is actually not going to be a good player to make lots of oxaloacetate because of where that equilibrium lies. So we're going to use pyruvate carboxylase to make oxaloacetate. And then again, we're going to see the use of malate dehydrogenase, but not in its traditional format. We're actually going to see the reverse of that reaction, the reverse of how it operates in the citric acid cycle. Oxaloacetate is going to be converted into malate, and why we have this is, remember, the only way to get malate, or the equivalent of oxaloacetate, out into the cytosol to be used in gluconeogenesis is to use the malate shuttle. So that's why we need to convert it into malate. Few steps then to get it out into the cytosol. Gluconeogenesis will convert it into pyruvate. So that is kind of one use of not siphoning oxaloacetate off of the pathway, but really siphoning malate off by reversing the malate dehydrogenase reaction. Kind of looking at the other side here, another option for pyruvate. Again, a cataplerotic pathway. Now we're going to be cannibalizing. Again, pyruvate converted to acetyl-CoA by pyruvate dehydrogenase, and then into citrate by citrate synthase. Citrate is an important metabolite that is siphoned off of the citric acid cycle. And again, that's something that because of where that equilibrium lies, we have plenty of citrate that we could siphon off. So pyruvate being converted into acetyl-CoA by pyruvate dehydrogenase, citrate synthase converts it into citrate. We do have a transporter out of the mitochondrion for citrate, and if you have citrate that accumulates in the cytosol, that is a signal for fatty acid synthesis. We'll get into the details of that in a few chapters, but kind of highlighting that it represents a cataplerotic reaction of the citric acid cycle. So a little bit more chemistry, sort of thinking about fates for pyruvate. So again, there's homework question 11 kind of talks about the different fates that we have for pyruvate, and now we've got 5 of them. So it's important to think about these branch point metabolites and the different chemistries that happen. So kind of highlighted on this figure is, again, one of the options for pyruvate is using pyruvate carboxylase to ultimately convert it into oxaloacetate, and that again can be siphoned off via malate to be used for gluconeogenesis. Pyruvate, again, chemistry with pyruvate dehydrogenase will make acetyl-CoA, and again that can be used for a variety of things in the citric acid cycle. So something that's new is thinking about the transaminase chemistry that we can have here. So I want to highlight up in blue here chemistry that happens with transaminases. So transaminases essentially swap a carbonyl group for a carbon-nitrogen bond. And again, we're not swapping these actual atoms in and out, but what we're doing is we're taking one molecule that has a carbonyl group and then an amino acid that has a carbon-nitrogen group. And we're essentially converting this guy into a carbonyl group and this guy into a carbon-nitrogen, which equates to taking something that is an alpha keto acid and making it into an amino acid, something that was an amino acid and converting it back into an alpha keto acid. So one of the things that'll become important as we move into our amino acid and our protein metabolism chapter, your amino acid structures aren't going away because you do need to be able to recognize what alpha keto acids correlate to what amino acids. So you essentially have to be able to take this carbonyl group, make it into a carbon-nitrogen bond, and then ask what's the amino acid. So that isn't going away, so we still have to know our amino acids for that. So another fate for pyruvate is this is why pyruvate and alanine are sort of interconvertible. We talked about this with our glycogen metabolism case study where we had a glycogen storage disease with Alex. And we talked about pyruvate can be easily converted into alanine and exported from tissues that are undergoing ischemic exercise. And again, the reason for that is it is just another way to handle that waste product of pyruvate by transamination chemistry that converts it into alanine. Alanine can then be exported. It can be converted back to glucose in the liver by this glucose-alanine cycle. So again, that's something we'll get into more details later on when we talk about some cycles to do with amino acids. One last little thing, just because it's on the slide here, to talk about this concept of reductive amination. So another way that we can interconvert alpha keto acids and amino acids is by this process of reductive amination. So again, we're going to be doing reduction chemistry, so we see NADH being oxidized here, and we actually use ammonium directly to add that carbon-nitrogen bond in there. So an important thing just to kind of introduce here, we'll see it later, we've got 20 amino acids, right? But when we talk about how we handle and metabolize amino acids, and we talk about needing to get rid of the nitrogen as an extra atom that's on those amino acids, one of the things that we're going to see here is we don't do that chemistry with all 20 amino acids. We basically do some chemistry to funnel amino acids to either aspartic acid or glutamic acid, and then we deal with kind of the nitrogen that are on those. So it's sort of a streamlining process, if you will, where we don't deal with nitrogen atoms on all 20. We do either transamination or kind of this deamination chemistry to funnel things to be these two amino acids, and then we deal with the nitrogen on those. So not something that's really important for you to know for this test and this learning unit on the citric acid cycle, but just kind of setting the stage for some chemistry that we're going to see in the future. All right, so a few other things that we talked about in this learning unit are the glyoxylate cycle, and then thinking about some important other functions of citric acid cycle chemistries. Okay, so one of the things that I have hopefully hammered into your brains is the fact that we as mammals cannot convert fats into glucose. And I shouldn't even say mammals, it's most animals cannot convert fats into glucose. And that's because fat breakdown generates acetyl-CoA, and acetyl-CoA is either committed to fats or to the citric acid cycle for conversion into energy. And so we do not have the ability to convert fats into glucose. However, if you are an organism that has a glyoxysome, which is an oxidative organelle that has some chemistries like the mitochondrion, then you are able to do what's called the glyoxylate cycle, and the net is the conversion of fats into glucose. So the net chemistry that happens in the glyoxysome is the conversion of fats to glucose. It happens in plants and in some bacteria. So I'm going to walk through the net of what I need you to know for this pathway. There's a structure, there's a diagram here that shows all the different steps. Again, we're not taking a deep dive into that, so I'm going to show you the parts that you need to know. All right, so we know triglycerides are fats, store fats. Triglycerides as fatty acids are broken down via beta oxidation into acetyl-CoA, and this process happens in the mitochondria. So again, we've got acetyl-CoA that's in the mitochondria. That acetyl-CoA is going to make its way into the glyoxysome. Now I'm overly simplifying this, but what I want you to know is that the net chemistry that happens in the glyoxysome is the conversion of 3 acetyl-CoA groups, so each acetyl-CoA being 2 carbons, so 3 of them is going to be converted into succinate and glyoxylate, the namesake of this cycle. So again, that's an oversimplification, but that's kind of the net of what happens, and then this glyoxylate continues in the cycle. This glyoxylate that's here is kind of like oxaloacetate. It's sort of generated and then reused. So it kind of is an intermediate, a catalyst, if you will, in this pathway. But the net chemistry that happens in the glyoxysome is that acetyl-CoA in the glyoxysome is converted to succinate. That 4-carbon piece exits the glyoxysome and goes into the mitochondria and utilizes 2 reactions that have no problem from a delta G standpoint. So it's going to use reactions 6 and 7, which happen favorably in the forward direction, ultimately converting succinate into malate. So it dips into the mitochondria for 2 chemistries. Again, we know we can export malate from the mitochondria to the cytosol because we've got the Malate Shuttle. Malate in the cytosol can now be converted into oxaloacetate by a cytosolic version of Malate Dehydrogenase. That oxaloacetate can then be used to make glucose in gluconeogenesis. So why is this important in plants, for example? Well, plants begin as seeds, right? Seeds are very rich in fats and oils. So we can take the oils and fats that are present in seeds and they can convert them essentially to glucose, which has uses either as starch for plant food or cellulose for plant growth. So again, this important pathway allows plants to store things very efficiently as fats, but then when needed, be able to convert them to glucose for growth. So again, highlighting something that we talked about last chapter, last learning unit with our gluconeogenesis chapter, or gluconeogenesis in our glycogen metabolism chapter. I'm going to revisit this because this is going to become important as we highlight an important citric acid cycle enzyme. So remember, we're talking about the malate shuttle. The malate shuttle works because we can convert oxaloacetate into malate in the mitochondrion, so we can transport it out and then reconvert it to oxaloacetate in the cytosol by using a cytosolic version of malate dehydrogenase. So again, malate dehydrogenase lives in 2 spots. So I'm going to spend a little bit of time talking about sort of the chemistry that can happen with malate dehydrogenase and kind of highlight this concept of substrate channeling and then metabolons. All right, so let's first here kind of define what a metabolon is. And we highlighted this is not metabolome or metabolomics. That's very different. That has to do with kind of looking at the net sum of whatever metabolites are present in an organism. A metabolon is defined as an association of enzymes from what often are sequential steps in a metabolic pathway. So if we think about these metabolic pathways sort of being like assembly lines, it makes sense to have sequential steps located next to one another in physical space. So by physically associating these sequential enzymes, we should make this process more efficient and importantly include and incorporate substrate channeling. So again, substrate channeling means the product of one enzymatic reaction is the substrate or the reactant for another one. And we see that beautifully highlighted here with malate dehydrogenase and citrate synthase. Okay, so remember, malate dehydrogenase is converted to oxaloacetate by malate dehydrogenase. This is a positive delta G, so we have very small concentrations of oxaloacetate that's being made by this process. So we kind of joked and said, oh, it's just this tiny little squirt of oxaloacetate that comes out. Well, if citrate synthase is actually connected right next to malate dehydrogenase, as soon as that little squirt of oxaloacetate comes out, it can take it and it can combine it with oxaloacetate, or I'm sorry, with acetyl-CoA, and it can make citrate. And in this process, I was just kind of reminding everybody that these 4 carbon atoms of oxaloacetate make these 4 carbon atoms of citrate, These are the two that come in from acetyl-CoA. This is the first CO2 that comes off. This is the second CO2 that comes off. So be able to think about that in terms of if I showed you the structure or had you draw the structure of citrate, identify where the parts come from, who's from oxaloacetate, who's from acetyl-CoA, and then where are the carbon atoms that come off as CO2, and when do they come off? Okay, so if we have citrate synthase that has a large negative delta G, kind of non-covalently associating very close to malate dehydrogenase, the tiny little squirt of oxaloacetate that can appear in the enzyme active site can be channeled easily over to the active site of citrate synthase, which, because of its large negative delta G, can quickly assemble that into citrate and then wait for the next little squirt of oxaloacetate to come out. So when malate dehydrogenase is doing citric acid cycle chemistry, it wants to be closely associated with citrate synthase so that it can have this effective substrate channeling. It can overcome the fact that we've got this positive delta G. Now, what I want to highlight is that's a different kind of chemistry that we want to have malate dehydrogenase do if it's helping to do the Malate-Aspartate Shuttle. All right, so remember, pyruvate can be converted into oxaloacetate by pyruvate carboxylase. So again, that's going to make oxaloacetate not by citric acid cycle, but another route. So we're going to have much higher concentrations of this. Well, if we're going to have this oxaloacetate be converted into malate, transported out to the cytosol, and then ultimately be made into glucose via gluconeogenesis, we need to have this malate dehydrogenase not be associated with citrate synthase. Remember, if malate dehydrogenase is closely associated with citrate synthase, it's going to be moving in the unfavorable but forward direction, squirting out a little bit of oxaloacetate so citrate synthase can use it. If we want citrate synthase to be working in the opposite direction, it needs to disassociate from citrate synthase so that it can do that chemistry in the reverse. It can take oxaloacetate and actually make it into malate. So part of the research that actually goes on with students in my group is we're looking at understanding what dictates this association between malate dehydrogenase and citrate synthase if we want to have chemistry going on, and then allows it to not be associated if we want to be doing different kinds of chemistry. So some really interesting things to think about what surface residues might be interacting or not interacting there for that process. Again, just to make sure I highlight so the chemistry is reviewed here from gluconeogenesis, once we get malate out into the cytosol, we're going to have malate dehydrogenase that's in its cytosolic form, kind of making oxaloacetate in the cytosol. Pepsi-K is still necessary to convert oxaloacetate into phosphenylpyruvate, and once we get to phosphenylpyruvate, reversing glycolysis via gluconeogenesis is pretty straightforward. The rest of the reactions, except for reactions 1 and 3 in glycolysis, which were pyruvate kinase and then PFK, those are the ones that we need to find different enzymes for. But other than that, the rest of the steps in gluconeogenesis are using the same enzymes as glycolysis, just running them in reverse. And that wraps up our understanding of chemistries with the citric acid cycle.